things always reminds me of, the tool different that we used to run on computers back in the day. And all these seats which are allocated all over the place. Okay, I think we can start. My name is Alessandro Pilotti. So I do not come from neither a bioinformatics nor an AI background. Okay. So you might think, okay, what am I doing here? So first, let me ask you a question. How many of you guys come from a biology background? Ah, that's a good one. How many of you have an AI background? Good. And how many of you have like a DevOps background? The rest of it. Okay, perfect. Yeah. Good. So this is what happened. At some point, I run a company which is called Cloud-Based Solutions. We are doing, we are involved in OpenStack since the Folsom release, so that means like 2012 approximately, right? And over the years, if you want, my typical skill consisted in being ahead of the curve. Meaning like, okay, I was doing OpenStack, it came relatively naturally, having quite some experience with all the VMware, Hyper-V and whatnot before. When it was time of container and Kubernetes, it was relatively natural. You knew everything that happened before. As a developer in that space, it was regularly easy to get into that and become very proficient, create products and solutions on top of that. And so on. So the whole, if you want, technological evolutions consisted in steps that were relatively easy to follow if you knew where the breadcrumbs were. Then came along AI. And I tried to tackle AI from a computational standpoint, like I did everything else. And while, of course, algorithms and everything clicked, I just couldn't really get the gist of it. And one day I realized, well, this is really not a computing problem, this is statistics and big data. And I was like, well, okay, I did some statistics doing like my computer science master a long, long time ago, right? But what about the rest? And then I realized, okay, where do I get the big data? And then I figured out that AI, unlike the technology I mentioned before, cannot really be addressed in an horizontal way. You can do Kubernetes and you don't care what type of workloads you run on top of it. You cannot do OpenStack and you can absolutely not care about what the VM do. For you, they're just black boxes. You just have to make sure they have proper computing, storage, and whatnot, right? And networking, and you're good. But AI, no. AI needs to be highly specialized, typically on various verticals. So I decided, okay, it's time to get back to academia. So I went to UCL, UK, and more precisely Burbank, because it's the perfect type of college if you're already somebody that works and everything. And I got what is called in the UK an MS, Master of Research, which is something in between a Master of Science and a PhD, let's say, on bioinformatics. Why bioinformatics? Because bioinformatics is a domain that has a lot of data, so something I needed to learn this. It's perfect ground for AI, because it's of course where a lot of AI is being researched, being applied. And I was really passionate about the subject, because molecular biology in the end is nothing else than coding living things instead of computers. So that's where my interest entered. Okay, so what I'm bring up here, and this is actually the first time I present this particular session because I just finished my master a couple of weeks ago, is actually how do you transition from decades and decades, I have more than 30 years of experience as a programmer and everything, to something completely different, and how do you approach AI and what results you can get, as somebody who's not in the field. And I learned quite a lot, so we'll try to get to this. Now, since this is a use case where I'm going to talk a lot about the type of problems I'm trying to solve, I will need to give you a little bit of an intro about molecular biology, more than macrobiology. And I will do that oversimplifying a lot of things. So for you guys in the domain, please take note that I'm trying to explain these things to people which do computers, right? At the same time, I limited that to 3 slides because I really don't want to hog the whole session, because you could go down a lot of rabbit holes when it comes to biology, right? I don't want to do that now. I want to go as soon as possible to the actual problems we're talking about. So the first thing we need here is DNA. You all know what DNA is, right? From a computing standpoint, it's not anything else than source code, which is in a quaternary base instead of a binary one. 4 bases, adenine, cytosine, threonine, guanine, right? Instead of 0 and 1s. And it's stored in the famous double helix, which is nothing else than a RAID 1 thing, right? We, in computers, if you have a server, you want to keep multiple copies, right? If you have one single copy, you cannot complain if you lose a disk. That's what happens in nature. That data is simply duplicated, and that prevents things, or mitigates in a way, the biological equivalent, for example, of bit rot and similar things, right? And then it's very complex space, right? But I just want to give you the basic things. Not all, so the usual thing that we think about when we think about DNA is that it's entirely a huge amount of source code. Well, it's not. I mean, in bacteria, the majority of it is what we call coding DNA, right? In eukaryotes, for example, mammals, humans, only a small percentage, around 2%, is actually coding. So the rest, it's a lot of data that has multiple functions, many of them we don't know them yet, right? Then, if that's the source code, how do we get to binaries, something that you equivalent to binary, something you want to execute, right? Well, we have an intermediate here. If you're familiar with Java, or even with Python, you're familiar with bytecode, which is an intermediate language, right? Which is between something like understandable by humans, source code, and the binary code itself. In nature, this is called messaging RNA, mRNA. You've probably heard it a lot, especially thanks to the COVID pandemic, mRNA vaccines and everything, right? mRNA is, to make it very simple, nothing else than a translated form of DNA. So you take a strand of DNA, and you get RNA, which is the same identical thing, with one difference, which is one of the bases. Threonine gets translated into yours, okay? So the T becomes U. So if you have a sequence like this one, T, G, G, T, A, and everything becomes U, G, G, A, N, U, N, okay? So this is the thing that gets then translated into the equivalent of the binary, of the executable things that we will talk in a second. There is a sort of compiler here, which is called RNA polymerase, okay? Which is a biological machine, which just takes the DNA, reads it, and spits out on the other end an RNA string, right? This is very interesting because the DNA, for example, in eukaryotes stays inside of the nucleus, while RNA gets sent out with complex mechanism in the cytoplasm or in the rest of the cell, where other things called ribosomes are in charge of taking that and generating nature's executables. Those executables are called proteins, right? Or polypeptides, more precisely, right? So, you guys for sure heard the term amino acids, right? There are 20 of them in nature that we use, right? And that's quite interesting. Those are the building blocks. Those are the Lego pieces, right? Or Tetris pieces, right? So nature uses a very specific number of those. And those are combined to create those executables used. The ribosome is this machine, which an incredible natural machine. Beautiful if you have time to read it. There is okay, I'm not going to go down the rabbit hole, but it's a very interesting thing. It's made by a mixture of RNA and proteins itself. It's made by a lot of subcomponents. Very, very interesting thing how these things evolve. You know. Anyway, this thing is very interesting because it takes a strain of mRNA and spits out on the other side proteins. Right. The very interesting thing here is that it takes always 3 bases from the RNA, which is called a codon, right? And generates on the other side for each of them one amino acid, right? Which chains together with the others, creating a polypeptide, right? Another very interesting thing is that the instruction table that translates those codons to proteins is almost the same for all living beings. extremely interesting. There are small differences, but it's basically a CPU table in which you take instructions and translate it into other. So it's an amazing thing that happens in it. Another interesting thing, okay, I'm not getting into the idea why there are 3 of them, but let's say more of them translate to the same protein, of course. The last one is a called wobble position, but okay, not anything there. A super interesting thing, if you're familiar with strings in C, for example, strings are null terminated. At the end of the string, there is a 0. Well, in nature, there is a particular set of colons, UA in terms of RNA, for example, which is called stop colon. So when the ribosome encounters the stop colons, it simply ends the protein. Like, it's literally a null terminated string, basically, right? Okay, so this is as much we need to know at the moment, but the important thing is that we go from strings of RNA bases, right, to strings of amino acids, which are these polypeptides. One or more polypeptides makes a protein, okay? So simple proteins are made by one single string of those. But getting towards the AI, as you can see, we are getting to strings, okay? So this is what we're going to deal with. Now, antibodies, talking about antibodies, because as you will see, the biological problem I solved is specific about antibodies. So antibodies are Y-shaped proteins. There are 2 copies of 2 chains, heavy and light. There is a constant and variable part. We care about the variable part. Why? Because that's the one that is largely in charge of binding with the pathogen inside of our body, right? There are 2 of them, heavy and light. They're relatively short, 110 to 130 amino acids, okay? They can be standardized in the same structures, right? They are composed by 7 regions, some of them more relevant for binding to antigens, so the enemies, if you want, inside of our body, the pathogens, and some of them more relevant at creating a structure to keep them together, right? Imagine them as Lego pieces meant to bind specifically with other Lego pieces, right? They specialize, they have a specific degree of affinity towards a specific antigen. So one type of antibody will develop inside the body for dealing with one specific type of pathogen, okay? So that's why probably you heard about IgG, IgM antibodies and everything, specific for, I don't know, SARS-CoV-2, and flu, and whatnot, right? The part of the antibody that binds with the antigen is called the part of. Okay? We need this because it's part of the problem. That's it, right? I summarized. People in biology, please don't kill me. Not only I'm not from the domain, but I also try to summarize it for people not in the domain without, hopefully, not boring too many people. So enough biology, back to computers. Now, how many of you heard about the transformer architecture? Good. How many of you use ChatGPT? Good. So all of you use that transformer architecture. Transformer is a big evolution, not a revolution, but an evolution in machine learning. It's a paper from 2017 called Attention is All You Need. Wonderful paper which introduced this model. This model then became ultra popular, of course, with the so-called large language models. Large language models are, of course, massive models, as the model implies, based on transformers and so on. We are going to use them now for biology problems. There is a difference. People use typically this type of models in NLP, natural language processing, meaning that you take natural language, that's like English, right, and you split it in pieces called tokens. So you need a tokenizer to do that. For example, here you have an example in which you have a string, antibodies are interesting proteins, split in tokens, which are antibodies are interesting proteins, right, in a dot. It doesn't have to be the whole word. Sometimes there are parts of words, you know, for example, and taking out the ing ending from, for example, interesting, right, and so on. So, but it depends on the so-called tokenizer. So tokenizer is an important thing because it splits it in pieces. Those pieces then will be passed to the model, and the model will work on that, as we will see later. In proteins, and also antibodies, we are not going to do that. We have one amino acid, one token. Right. I didn't mention it before. Each amino acid has a letter that identifies it. Okay, very simple. Now, in the project I did for my master, I took some so-called protein language models, PLMs, right, and some ABLMs, which are pre-existing. PLMs are trained on generic proteins. They are massive models. ESN2, for example, is one done by Meta, like Facebook, to say so. You might have heard of AlphaFold, a project by Google, which also uses this type of methodology, also has a different architecture though. And then we're going to use antibody language models, which are trained only on antibodies. Okay. Here is a list of them: anti-BERT, anti-BERTA2, ABLang2, FON, FTSM. Doesn't matter for this context. They are all based indirectly on the model architecture called BERT, made by Google, which is an application of the transformer model. Okay. Now, after this introduction, here is the problem we are looking at. Again, this is a use case, so I'm presenting you what I did here, but I think it can be generalized to a lot of other problems in biology and outside of biology where you need to deal with AI, especially when it comes to something which is not just using existing, like running prediction against ChatGPT or existing models, in which you want actually to train your own or fine-tune your own as a heuristic. So two problems. In one, we want to assess antigen specificity, meaning, given an antibody sequence, we want to know if that will bind or not with a given antigen. We did that with big databases of SARS-CoV-2 spike proteins, right? You're probably all familiar with that, right? One unwanted side effect of this is that at the next pandemic I would be able to say, I did my own research. But let's say I didn't want to go and study specifically COVID, right? It's just something that it's very easy to study, meaning that unfortunately we have to do a lot of research in that space, meaning that we have a lot of databases of antibodies, meaning donors basically to blood, we sequence those and we know how they look like. And we do here, so this is a binary classification problem, meaning that we want to do yes and no. So given that sequence, you tell me, yes, it binds, no, it doesn't bind. The other problem is much more complicated, it's paratope prediction. Given an antibody sequence, which positions belong or not to the paratope. This is token classification, meaning for each token, for each position, we want to find which one it is. Okay. Fine-tuning models requires 3 data sets, at least 2, let's say, 3 at least. Training, evaluation, testing. Not entering into details here, but generally speaking, you want to have a full data set of labeled sequences. In our case, for example, for the first problem, that would be sequences of antibodies, and for each of them we have a label like it binds, it doesn't. You take them by getting, for example, a lot of pre-COVID sequences which are all not binding, and you take, for example, data from people that did vaccines, people that got the illness, and so on. And then you have test evaluation that you will need to optimize your model as it runs. And then you have another one which is for testing, which is to be completely separated to make sure that it does not happen. In the process, you need to do a lot of so-called hyperparameter optimization, in which you want to get to the point in which your model works in the best possible way. Right? This works by optimizing epochs, basically runs that you do on the data, batch size, how many sequences you pass in at a time, right? Things like this. Okay, we'll get back to that. One important topic is data leakage. When you see a model which has more than 90% performance, you have to be very suspicious, because it's too good to be true. Sometimes it could be really good, maybe they did a fantastic job, but you want to take a deep look into it. The reason for this is that very often, if you have train test contamination, meaning that information which is within the test data set belongs also to the train data set, is like a student that knows the answers when you do an exam, right? It's not cheating, but gets close, right? So you want to make sure that the model doesn't know the answer when you do that. This is very difficult, especially for antibodies. I'm not getting into the topic right now, but for example, we kept completely different donors, and we clustered the data to make sure that only different antibodies were used in the two domains. Okay? That means you see where verticalization comes to hand. If you work on any other domain problem, you need to try to understand what makes your data unique and make sure your model is not cheating with it. Now, getting to the core of the automation part here, this whole project ended up in around 600 tasks. You are not going to run 600 tasks manually, right? So you want to run them in a sort of pipeline. So a big part of my work here was writing actually a pipeline, thankfully, because that's actually what I do for a living. So getting the biology side of things was important. This can be done with directed acyclic graphs, DAGs. Ever heard of DAGs? Good. Apache Airflow is one of them, but there are so many others. There is Dagger, there is Soda Project. Even in biology, there are them. And then once you sequentialize, you have to decide what you use to run those things. As a DevOps, Kubernetes was the obvious choice here, right? It takes a lot of ground also in the HPC space. But there you will find also a lot of Slurm, a lot of SGE, of the things. Here's a quick outlook about how the models look like. I'm not getting into this, we don't really have the time for it, but you have a lot of data preparation. You are clustering the training data set, you're splitting training validation, you cluster the test data set, you undersample it if needed, meaning you balance the classes, that you don't want to have too many zeros and ones, right? Compare. You want to shuffle also the labels because you want to see also how the models works and completely randomize data, right? Because with randomizing, you want to have a 0.5 performance, like random. And then you feed that to a bunch of tasks. Fine-tuning, the ones in yellow are GPU tasks, okay? So fine-tuning, prediction, fetching attention weights, running work on that, running the same things on the pre-trained models, and so on, right? At the end of the whole story, all this data gets into some reports, the reports get processed, and an email eventually arrives, right? The whole thing, I'm running these things on two Nvidia H100s with NVLink. It takes around 15 hours to do the whole thing, right? So you want to make it as automatic as you can. And this is the other one for Paratalk prediction for the other project. Not entering into details, but you can share happily the slides, and if you have questions, I will be more than happy to share them afterwards. Let me start a demo while I talk about the last aspects here. So this is Apache Airflow. It has a very nice UI. These are my two pipelines. I take the sequence classification one. Hopefully the network here will help us. Yes, it does. And here you see, oh, they have only a few tasks. But then I expand them. Network, yes. And as you can see, there are a lot of them, right? So those are all the tasks which are running. To make things optimal, I made it in a way that, like a make file, it will check if it has to run. For example, if when fine-tuning a model is needed, it's checked by saying, hey, is the input data newer than the model I created? If yes, I run the whole fine-tuning, which takes a long time. If not, I don't, right? And this has to be done on a lot of models, so you really don't want to waste GPU resources for that. And you can also show them in a graph here. So there are, as you can see, a lot of them. They're all connected, and you get at the end your nice little result. Okay. Now, what I did right now, I prepared it in a way in which we are only run one single prediction, because we don't have the time, of course, to run the whole thing, right? So I just click run. This triggers the whole thing. And I can look, of course, at the various results. Or I can also you're familiar with KNNS? It's a wonderful tool, right, for So at some point, as it runs, you will see here containers popping up. The pink ones are skipped. The green ones are the ones being executed. As it takes a bit to get there, get back to the slides, since we don't have too much time and we get there in a moment. Now, did I use OpenStack plus Kubernetes or just bare metal Kubernetes? Two advantages from both sides. OpenStack plus Kubernetes is fantastic if you have something more than just containers. So if you have a mixture of VMs and containers is particularly good. It offers great flexibility in managing Kubernetes cluster, multi-tenancy out of the box. CPU access is typically with Nvidia MIG. Negative sides, MIG allows one single GPU per workload. I mean, you can pass more than one, but it will run out. And it's one extra layer for manage. On the other side, bare metal Kubernetes, direct access to everything. You can decide to use whether time slicing, MPS, MIG, whatever. Less day 2 ops complexity. Multi-tenancy is harder to achieve and limited elasticity, right? Familiar with MIG, time slicing, and MPS, by any chance? Any of you? Great. So, there are three ways to share GPUs. Nvidia GPUs specifically. MIG is the way in which you partition them at the hardware level and you pass them basically to every workload. Ideal for isolation, especially if your tenants don't trust each other. Time slicing, perfect if you're the only owner, of course, of the user of that infrastructure, and it's the most tried and true from the three of them. Usually no problems whatsoever. On the other side, you have no isolation chance. On the other side, the problem is, of course, performance is not ideal. There is a lot of overhead in this constant round-robin type of context switching. MPS is newer, less overhead. It's a little bit in between MIG and time slicing. Problem is that it's still experimental in the Kate's device plugin. So your mileage may vary in usage, right? Good. Optimizations. All these models use either PyTorch or accelerate behind the scenes. We want to use either distribute data parallel, DDP, not very efficient because it copies the whole model all the time. My biggest model here has 15 billion parameters. It is not going to fit in 100 gigs of RAM in a single H100. While if I split it in two and I slice it, it works. To do that, you can use either FDSP or, much better, deep speed. Deep speed was created by Microsoft, wonderful tool. It's a magic thing. You just enable it and it will do the work for you. Okay. It uses zero redundancy optimization, which comes in three possible options, 1, 2, 3. The general suggestion is like, if you can, just go filter. Okay. So the main idea, especially with the last one, is that the whole model will be split and divided. So you have slices of the model across all GPUs. So if this model is massive, and you have GPUs, for example, linked either via NVLink or with networking, right, across multiple hosts, you can do that better, right? You're basically scaling wonderfully, right? And the rest of the memory that is most available, you can leave it and pass it through your other workloads, which are running in parallel, right? Because remember, you're hammering your GPUs with a ton of containers, which all do GPU works at the same time. Okay, here are my repositories. So one of them contains all the source code. This is, again, highly specific for what I'm doing here, right? But I think it can be used and generalized for other stuff. Then you have one repository with a definition of the DAGs, so meaning the workflows, etc. And so on. So the first repository are all the building blocks, like fine-tuning, prediction, and so on. The second one is putting all the things together. And then there is a last one, which has basically Docker files. There is one Docker file with a base CUDA image for everything which is Python CUDA related, right? And then a separate with R R is a language used typically for statistics. So R is one of the, I added one more language to my arsenal if you want here. And I wrote all the reports with that. Okay, demo time. Oh, that was set, checking the demo finishing. Okay, there is still one container running here. So this is running my R work, right? So I'm multitasking here because we have only half an hour for a lot of content, right? So, and it's almost finished. And if I look here, you see all my things are running. It should be almost done. Updating networking is not particularly fast today, so you see everything is either ping or executed, and I have this one running. I can click on it. I can click on the logs, and I can see the same process. So the same output that you saw from the log from the container, you can see here. Almost there, come on. So I can show you also the results, and then get that. Okay. At the end of it, there is one last task which will send out the emails, and then that's it. Any questions when we wait?
That's very interesting. Did you make any comparison between running in Kubernetes versus Slurm or SGE in terms of the execution time?
Yes. Well, the good thing with Kubernetes, and in general, all these various projects, right, infrastructural, let's say, management tools, if you want, they have a role, which is the one to schedule resources, right? But once you have access to the resources, you have full control of them, right? If you say, I need 2 GPUs with this amount of, I don't know, memory and whatnot, IOPS and whatnot, you get that amount. So once that is obtained, they are all relatively similar, with one distinction that we all know that Kubernetes running container, it's a bit fuzzy, of course, the allocation, right? You put limits, but it's a bit more difficult, of course, to optimize how this distribution happens, right? But it can be, there are a lot of tips and tricks, of course, there to work that out, you know. The important thing is the quality of the scheduling, right? Kubernetes is not a scheduler per se. The scheduling is actually delegated to Apache Airflow in this case. So Slurm, it's better from a scheduling perspective. It also uses some sort of containers, but it's significantly worse in terms of developer experience, especially for people like us, which know Docker very well and have to learn something else. SGE, on the other side, is a drama because it's an extremely legacy type of thing. You find it often, for example, they have it there at UCL, right? And I made actually a wrapper, so this thing works not only with Kubernetes, it works also with SGE. You just say, hey, where do you want me to run it? We can distribute it with multiple of them, right? So there, you have basically, you pass in just a bash script, and then you just have to hope that things will work. Right? This is the problematic thing, right? Okay, it's finished, meaning that I got my email. And just a moment to find the email. Here it is. And here I will have my metrics. So here's all my R code, and it takes a while to load. And here you see there are a lot of them as the email will load. You see, those are all performance results. And then there is another one here. Tension comparison, why is the network so slow? Come on, okay. I'm not entering into the details here, but the general story is like, you see how the performance of the model improves on specific areas of the antibodies called CDRs, which are the ones relevant. So if you see higher spikes, it means that it's actually able to understand that there is actually where the antigen is bound. So your COVID, if you want, your SARS-CoV-2 molecule is actually being bound to the antibody, right? Okay, so I think that ends the demo, but let's go out with a couple of questions. I think we have time.
So you're running this across 2 M100s?
Yeah.
H100s.
Yeah.
Yeah. Why do you need the infrastructure stack if you're just going to run Airflow?
Yeah, surely. But the infrastructure side, there are two of them. It could be OpenStack plus Kubernetes, right? But in the one that I showed you right now, it's a basic Kubernetes deployment, right? With K3S in this case, for demo standpoint, but it could be anything else, right? And I have as I mentioned before, the plugin. Let me see if I can solve it here. Show you some things. So, for example, time slicing values. So here there's a relatively time slicing configuration. And I also have the equivalent for MIG and NPS, right? So it's simply splitting each GPU in 10 slices. So I have 20 slices in total that gets assigned. I use NFS for data sharing. So there is an FS, there is a PV and a PVC that get shared across the model, so they all share the same thing. I could use SFFS, of course, for this, in a larger model, right? This is largely a research, single server type of project, right? But it could be scaled down. If you want, we can talk a bit longer. I think it will take me half an hour just to go through the whole infrastructure. Yeah. Any other quick question?
Did you consider adopting Kubeflow over the Kubernetes?
Wonderful question. The reason I didn't is connected with the previous question, because I need this to work with SG, with Slurm, and a lot of other things, so I couldn't just do Kubernetes. Behind, I think?
It was just the same question.
Okay.
And final consideration, binding is not really a binary problem, right? You have to set a threshold. It's an energy problem.
Wonderful question. So if you look, then we go more into biology, but
Oh yeah, forget. I'll not talk to us.
okay.
Forget
Okay,
it.
okay. As you can see, there is a
Wonderful talking.
in AI, nothing is 0-1. Everything is like in between somewhere, right? Okay, I think we're a bit over time, so thank you very much, right? If you have more questions.